Okay, can you hear me, and see my slide?
Yes.
Okay, fantastic. Great. Thank you, Guy and Tom, for organizing this online workshop. It's been a lot of fun. So as Guy mentioned, I have a somewhat unusual title, perhaps. So 9 months, 2 days, 9 years and 2 months, 16 days to go. So why that title? So on the 15th of June, 2010, I gave a talk at the second IWBDA, International Workshop on Biodesign Automation. And I declared at that time that I wanted to see the creation of a fully programmed nanoscale machine with all the components specified and designed by engineers, so no magic components. And that I wanted to see this, done within 20 years, which is roughly when I'm planning on retiring. And so that's sort of what set the parameters. And so I've been keeping track, and this is now the amount of time left to go. So 9 years, 2 months, and 16 days. And so I wanted to tell you a little bit about sort of where I think things are at, more broadly than just what's going on in my group, of course, and what some of the challenges are. So what I'd like to do is to try and justify the following claims, which some of you have seen before. And that last time you saw it, I probably said we're within 10 to 15 years. That's because I gave a talk on this about a year ago. So I think we're within 9 to 15 years of being able to produce genetically programmed synthetic cells in multicellular machines. But there's a lot of work to be done. And I think we're going to need to move from talking about components to circuits, which you just saw, for example, in Domitila's talk, to really thinking about subsystems and systems. And as we put things together, how do we do that? And we see this in other engineering disciplines, and I think we're going to have to see it here as well. The other thing I'd say is that synthetic biologists, for example, Domitila's work was putting biological circuits into living cells. I guess I've come over the last decade to feel like synthetic cells, that is non-living systems, are a more promising approach to synthetic biology generally than cell-based methods. And so I've kind of, moved in that direction. I think, not inconsistent at all with the goals of the workshop and the things that Tom talked about yesterday and that you heard about in other talks. And so I'll see whether or not I convince you of that point of view by the end of this talk. Okay, so this is small on number of slides, but it'll take me probably a half hour to go through it all. So what I'd like to do is tell you a little bit about what I think it's going to take to make things like this happen. And then I'll give you a couple of examples of things that we've tried that have mainly not worked and where we need help. So try and say, look, here's some stuff that's not working that I think more people need to be working on and helping solve. Some of them line up with some things that you've heard about prior to this. And hopefully we'll hear about more tomorrow. Some of them don't, but I hope it at least motivates people. And then I'll say a little bit about some work that we've been doing in modeling around the idea of bringing together subsystems and other things. And so I'll tell you about some of that work. And then at the end, which will mainly happen, I think, in the breakouts, we can kind of discuss and debate, and you can tell me about things that are going on that can contribute to this, I hope. Okay, so with that then, let me describe a little bit about a synthetic cell. So this is my picture of a synthetic cell. It looks very much like a picture in the book with Domitila of a natural cell, which is not too surprising, except that I've sort of broken it up into the various subsystems that I think need to be present if we're going to build synthetic cells. And so I like to think of a synthetic cell like we might think of an airplane or a cell phone or the power grid. That is, it's a bunch of different subsystems that work together to carry out some function. And each of these subsystems has its own purpose. So there's, of course, some sort of spatial organization, the cell wall and internal structure. This might be something that's made out of DNA origami or made out of, different types of DNA nanotechnology. The types of things that Peng described yesterday, for example, could be the way that we create this chassis, and it's got, kind of internal structure to it. But we need some way to spatially organize things, I believe. We need some way to power the system, so some sort of a metabolic subsystem. Think of this as, plugging something into the wall or, wirelessly charging it or exposing it to sunlight or chemicals or whatever. We certainly need to be able to sense and signal, that is basically get information into and out of whatever our chassis is. And so some mechanism for doing that. Inside the cell, we need regulation and computation, and this is actually the place I think where we're most mature as a field, but also in terms of things that we've done in my group, in terms of thinking about different ways to regulate things. Again, Domitila's talk's a great example of that. But also computation, various types of gates and other things using genelets or whatever. So, I mean, you've sort of heard about various things going on. And I should say what's going on the inside may look very different than the way that biology does it. There may not be proteins, right? It may just be DNA and RNA, for example. And I think that's fine. Again, I think, whatever those subsystems are and whatever kind of technology they're based on, we want to think about how we integrate that with the other pieces of things. And finally, we presumably want these to do something. And so there needs to be some actuation mechanism. I think there are lots of different types of actuation. We can have chemical actuation, that is, we can export molecules in the environment around us. These might be therapeutics or things that help plants grow, or sort of things that help wounds heal, or whatever it happens to be. But they may also be things that exert force, either by allowing things to move around or by resisting motion or other things. And so here on the actuation, you can sort of think of that as both the flagellar mechanism over on the left, but also some of the export mechanisms we might have for getting small molecules out into the environment that are not for communication per se, but are to actually act on the environment. So I think all of these things are subsystems that need to exist. There may be more. These are the 5 primary ones that I sort of focus on thinking about putting together into a working system. Now, in order to do that, I think that there are things that are going to be needed to have a design workflow that we use for doing this. I think we're going to need, and these are things that we and many others have worked on, I think we're going to need simplified environments for prototyping and characterization of the subsystems. That is, you're going to need to be able to build up that metabolic subsystem without having to build the entire system, or build up that sensing and communication subsystem without having to have all of the other parts present. So think of that as a prototyping environment. We're going to need modeling and analysis tools for design verification, and as I'll talk about later, specification. And of course, we're going to need test environments that allow us to actually test and make sure things are working. So before we deliver them to a system integrator, we can say, yes, we satisfy the specifications for this component. And these would, of course, be things that we would see in other engineering disciplines. Again, think cell phones, airplanes, cars, power grid, all of the things, big, large, complicated engineering systems around us. And one of the things I think that's important is that we have to recognize this is not going to happen within a single group of a dozen graduate students and postdocs working together, right? This is thousands of people working, right? The number of engineers it takes to build an airplane, a cell phone, the power grid, et cetera, are thousands across different organizations, right? Each with their own specialty, and system integration being its own specialty. And so I think we have to start thinking that way in the context of synthetic biology and in the context of synthetic cells. I will point out that some of the elements of this design workflow are useful for their own purposes, right? And so, this may not be something that we just apply to synthetic cells, and so it may come up in other places. And once we have this notion of, kind of the unit component, right, the cell, we can, of course, start thinking about things like multiple synthetic cells and having things operating together. And, one of the examples that I like to use is, wouldn't it be great if engineers could design and build a machine that extracted chemicals from the environment around it, transported those chemicals and processed them to put them into sort of different forms, and then packaged them in a way that was particularly convenient for use by humans to do something interesting. And of course, a great natural example of that's an orange tree, right? I mean, it's exactly what an orange tree does. And so, I think it'd be great to be able to go design and build an orange tree, right? What's it going to take to do that? That's not 20 years from now. That's probably a little bit longer, but I think some of the first steps of that we can do. And of course, if we can build a machine that, right, can extract chemicals out of the ground and create oranges, there's no reason that it needs to just create oranges, right? We could create different fruits on the same, out of the same machine. And so we can think about what do we need to do in order to design and build such things. So to me, this is really what I'd love to see us, kind of community people working on this doing. Again, I'm agnostic in terms of what the underlying technologies are that go into all of this, but I do think we need to start taking a systems and subsystems, if you want, point of view to this. Now, this is not, novel. Lots of people think about this. Many people have already sort of said this in the context of artificial cells and other things. I think the articulation I'm giving here has come out of a series of conversations over about the past 3 years with one particular group that's been doing this, and that's the Build-A-Cell Consortium here in the US. And so if you're not familiar with this, I'd encourage you to go to that URL and find out more about it. It's a basically group of people who are interested in doing this. We have no money attached to this, a little bit for organizing meetings and other things, but we get together every 6 months. We've taken a break during the pandemic. We had one remote meeting and then decided we'd wait for the next one to be in person. But this is something that a group, it's similar to the Fabricell group in the UK in some sense, but our version of it here in the US that we've been building up is called Build-A-Cell. And everything that I'm talking about, you can find pieces of on the Build a Cell website, including all the repositories for code and things like that. Okay, so that's sort of big picture. I'm going to talk about a couple of different pieces of all of this, and not everything, but quite a few of the pieces. I'll basically talk about the metabolic subsystem, briefly tell you what we're doing, what's working, what's not working, and what the challenges are. Then I'll talk about things that embed in the membrane and allow us to interact with the outside world. So that's sensing, signaling, actuation, things of that sort. And then I'll say a little bit, probably the most, about modeling and analysis tools. Okay, so just starting on the metabolic subsystem, this is, of course, the part that's supposed to power the insides, right? And we do a lot of work in cell-free systems using TXTL and various cell-free extracts or pure express or other things. And the problem with these is that they run for a very limited period of time before you run out of resources, right? Sort of 4 to 6 hours is a kind of typical number. And so if we're going to have synthetic cells that do something that take more than 4 to 6 hours, this is going to take a little while. And so we need to somehow power the system, right? Somehow get energy into the system and provide whatever's needed. We may also need raw materials, right? So we may or may not decide that the synthetic cells should synthesize NTPs, which is, of course, what a natural cell would do. We might want to just import the NTPs from the outside. That's a perfectly fine way to do it as well. So I should say the work we've been doing here is the work of an undergraduate in my lab, Ankita Roy-Choudhury, and she is actually going to Peng's lab starting in the fall, and we'll be working there. So there's nice connections with things that we've already heard a little bit about. So maybe she'll take some of these ideas with her and maybe bring back some of Peng's ideas into how we can incorporate those into the broader notion of a synthetic cell. Okay, so we've done things in cell-free systems with metabolic pathways. It's a very effective environment for prototyping metabolic pathways. This is an example of a 1,4-BDO pathway that we did work on a number of years ago. One of my graduate students, Yang Wang, did this, and Yang Wu, sorry, and she demonstrated the ability to kind of implement a metabolic pathway in cell-free and to prototype very quickly, but to change the concentrations of different enzymes and other things. And so we can imagine building relatively complex pathways on the inside. With Ankita, we then started to say, okay, great. One of the issues is you run out of ATP very quickly, and so we'd like to do some sort of ATP regeneration. And so we started by trying to think about an ATP rheostat that Jim Bowie had designed at UCLA. I sort of showed a picture of a piece of it here, kind of flipped on its side. And it's a complex pathway, just like the ones we've done before. But this number of enzymes and other things, it's not impossible. It's getting to be big, but we think we can do that. But I have to say that we started looking at this and we were kind of optimistic that we could go implement this and use it to regenerate ATP and see longer life. But we got stuck by COVID, of course, as all of us did. And so we went from doing things experimentally or thinking about doing things experimentally to doing things with models. And I'll tell you more about those tools in a bit. And, just the amount of ATP regeneration and things we could get and the difficulty in getting this to actually extend the life of reactions led us to sort of wonder whether this was the right approach, right? Should we really be regenerating ATP? And so we've sort of backed away from this and are now looking at things where we're just trying to import things. Can we build importers? So we tend to use phospholipid vesicles as our mechanism for generating a chassis. Doesn't have to be that, but that's what we use. And so in that bilipid layer, we can embed transport proteins, and I'll say more about that in a second. And so we've been sort of looking at maybe we can just import the things that we need to import in order to do that. So there are lots of challenges in this area. I'd say we haven't figured out something that works. We not yet figured out how to get reactions to run for, say, days at a time inside of a phospholipid vesicle. We would like to be able to do that. So I think these are very much open questions, right? How can we provide continuous conversion or continuous input? What sources of power should we utilize? Is it sugar? Is it mechanical, electrical energy, light, other things? Do we have to recreate a minimal metabolism in order to get a synthetic cell booted up? Or can we use the outside world? I mean, after all, your cell phone doesn't sort of generate energy from natural gas or coal, right? You get it from electricity, right? So it's all converted someplace else and then you just import it in. Maybe we need to take a similar attitude. All right, so on the metabolic subsystem, we need something. We don't have anything. We haven't found things that work yet, right? We're still trying. Ankita's, graduating in another couple of months. We'll see, how far we can get. We'll start up the next student after that and see what we can do. All right, another area or class of areas that we're interested in is the sensing and actuation subsystems. And here we're really talking about sort of bringing molecules in, sending molecules out, right, as a particular piece of that, but also actuating motion, right? And so implementing things like protoflagellar motors, and this is actually work that ties back to Imperial, started with some conversations with Tom Ellis, but also Oscar Says, who talked earlier, and Yuval Alani, really trying to think about what can be done. The thing that all of these have in common is that they are membrane-bound proteins, right? And so we have to think about things that embed and are functional inside the membrane, and we would like these to be controllable in some way. So we have done work on membrane-bound proteins in cell-free systems before. So this is work of Xiaobin Guo, who's now in China, and the university, and looking at nanodiscs. So these are basically bilipids, so lipids that get kind of put inside a membrane scaffold protein, MSP, and held into a little disc. And you can use them to embed proteins and explore and prototype and other things. But of course, there's no inside and outside on a nanodisc, right? It's in a solution, and so you can't have an inside and outside. So we'd like to be able to do this inside vesicles. And so Zoila Jurado in my group has been working on doing this with MSBA. So MSBA is a membrane-bound transporter that uses ATP to bring things in, or will bring things in or send things out, depending on which way you orient it. And so we're thinking about how can we use MSBA as a mechanism for controlling export and import. It also has some interesting properties. It actually interacts with the bilipid layer in a really interesting way and flips some lipids around and other things. And we're exploiting that in various ways. And so, but I would say it's been a struggle, right? So getting things that embed and are functional in the membrane. So people have done reconstitution experiments before. We're sort of interested in expressing these inside, so DNA inside the vesicle that expresses this protein, right? And then allows it to embed. And that's been a real challenge to get to work. And then furthermore, we want it to be controllable, right? We don't just want it to be a transporter that always transports. We want to be able to turn it on and off so that it's a controlled mechanism for either export or import of molecules. We've also been looking, and again, this started out with some of the discussions with Oscar and Yuval at these ATP synthases. So you actually saw this, I think it was on Oscar's talk, maybe it was on Tom's, I can't remember, but one of the two of you showed this. This is an ATP synthase motor. So this is a very complicated non-equilibrium device in which ATP gets converted to ADP and causes a motor to spin. And people have actually reconstituted this and shown the ability to spin actin filaments. So we are now working, and this is the work of Manisha Kapaswala, on embedding that into a vesicle and trying to get the vesicle to move. And at least our calculations show that with the speeds and amount of forces and other things we can generate, this should actually work. And so this is all ongoing stuff. I would say that the getting things to embed has been hard. We're still trying. If anybody's working on embedding proteins, especially those expressed inside the vesicle, and has some ideas for us, please let us know. And as I said, we are trying to move from reconstituted things, which people have done, to really expressing inside and making things be controllable. So lots of challenges here. We have a lot of problems just getting things to work consistently. So I think that some of these are just experimental issues. Figuring out whether things are actually functioning or not is hard at the molecular level. So what are the assays that you use? You don't want to build the whole system and say, look, it moves, right? We need kind of preliminary assays before that. And as we've gotten into the modeling side of this, the molecular scale effects of what's going on and how the proteins interact with the membrane, et cetera, has been very interesting. Some of it is known and understood, but much of it isn't. And so what level of detail we need in the models and other things is something that we're working on. Okay, so that's sort of on the experimental side, things that we're trying. What I want to mainly talk about for the next 5 minutes or so is more on the modeling side of things. So let me start by just saying a couple of things about models generally. And this is something that was very nicely articulated in a book that Edward Lee wrote called Plato and the Nerd, about the role of modeling and how models differ in sciences and engineering. And so I've sort of written it as 4 different levels of modeling that you can do. The first level of modeling is that you want to show that I can build a model that captures what I measure, right? There's some experimental effect. I have some explanation for why this works. And I want to show that I can make a mathematical model that basically matches my results on my experiment and therefore validates, right, my proposed method by which this works, right? So what's happening is that we're getting resource limits and, right, sort of these co-activators are causing, different effects to happen. And when I build a model for that, indeed, I see that I can get a model that's plausible. Um, and all I'm really trying to do is match data and show that the mechanisms that I'm saying are responsible could be responsible because I can find parameters and models that show the same thing as I got with my experiments. I call that kind of level zero model. The next thing one would like to do that you start to get more to engineering is to make a prediction, right? Fine, if I want it to do better, what should I do, right? How should I modify it? And again, you saw this nicely, I think, in the work that Domitila is doing, where now you can take that model and say, well, which gain should I crank up, right? Or, which components need to be there? And if I do that, what will I see? So this is a different model. I'm not trying to describe data that I already have, but rather make a prediction and use that prediction to see what goes on. The next layer is to make, not decisions, but design decisions, right? And this really more at the architecture level, right? So now I'd like to design a controller, I'd like to design an overall system. And what's interesting is that if I just want to make a prediction, I've got a model and I'm going to change these parameters of what's going to happen, the level of detail might be different than if I want to make a design decision. Should I do A or B may not require as precise a model, or it might require a more precise model. And as those of you who do modeling know, you have to know what question you're trying to answer in order to decide what model you want to build. The last type of model, which I call a level 3 model, is I think where we need to be heading. And that is, and this is one of the comments that Ed Lee makes, that models are our description of the system, of the behavior we are trying to design. Right? That when we say, here's a transfer function in a linear system sense, right? Input time series to output time series that I want my amplifier to look like, or that I want my airplane to look like, or whatever. I'm actually specifying the desired behavior, and then I design the system to match that behavior, right? So it's a little bit different than making design decisions. I'm actually telling you, here's what your system should do, here's how it should interact when you interconnect. And so models as a specification of behavior, I think, is important, and that's a place I think we need to head. Okay, so this is just kind of high level stuff about modeling. We've been developing a number of tools over the years for doing modeling and building into the design build test cycle. I've sort of shown them here. I'm going to talk a little bit about Biocompiler, sub-SBML, and Bioscrape, mainly about Biocompiler. But these tools are intended to kind of fit together and help use things. It's also, I think, important to use other tools that other people are developing, and there are a lot of them. One of the ones that we've kind of come into contact with and I think has some great utility for us is one called Vivarium that's coming out of Marcus Covert's lab at Stanford, and we're working with him to integrate his group to integrate Vivarium into some of the things that we're doing. Okay, so just sort of, there's lots of little different tools out there. These are all Python-based tools, open source. You can find them on the Build-A-Cell website, et cetera. I want to talk mainly about Biocompiler. And so what is Biocompiler? So as the name implies, it is a compiler for chemical reaction networks. That is, it takes a higher level description of some desired circuit and then gives you a chemical reaction network. It compiles to a chemical reaction network. And so the block diagram here shows the elements of it. And the basic idea is that I want to combine mixtures, components, mechanisms, and parameters to get a bunch of species and reactions that eventually become a chemical reaction network. So what is a mixture? The mixture is, am I inside of a synthetic cell? Am I inside an E. coli? Am I, sort of in a well-stirred system? Am I, in a mammalian cell? Each of these has different chemical contexts, right? And you want to sort of say the same components. I can put the same DNA in all of those systems and I'll get a different answer. The mixture, right, is what's determining that different answer. What's the ribosome concentration? What's the RNA polymerase concentration? How many NTPs do I have? Things like that. Those go in the mixture. A component is a functional part, right? This might be something that is a set of DNA that is going to translate or be transcribed into RNA polymerase that is going to be translated into a protein and then fold. And so I have a bunch of components to go along with that. If we want to build a model of a repressolator, we want to build a feed-forward networks that Domitila talked about, et cetera, there are a number of components there. The mechanism part is sort of what I think of as being kind of special about Biocompiler versus other things. The mechanism is the model that we use to describe the processes that implement that component. So for example, transcription, right, is a process. And so the mechanism of transcription has a model that goes with it. And one of the things that we do within Biocompiler is that we have multiple different mechanisms that we can use for standard biochemical processes. So for example, we might model transcription and translation just using a Hill function with some delays, but we might model transcription and translation at a little more detail. DNA binds to protein, that creates mRNA, and then that creates the protein, right? And so that's maybe a set of simple reactions. Or we might really go whole hog, right, and talk about RNA polymerase binds. There's an isomerization mechanism by which you go from a closed complex to an open complex. Then you transcribe, right, sort of base pair by base pair, et cetera. The mechanism determines what species and reactions we need as we do all of that. And once you have all of those species and reactions plus everything else, right, you can finally put together your compiled chemical reaction network. And so, for example, just to sort of add on that, right, I might have a very simple model, right, for transcription, right. So this is just a differential equation model with repression, right. That's the Hill function representation. Or I might say, no, I can get that same model, but in more detail by saying that RNA polymerase binds to DNA polymerase. That creates a complex. That complex then creates mRNA. And if I have a repressor there, right, it sort of blocks the DNA so that it can't happen. And I've written translation down there. That's a different model, more species involved, right? Because now I've got to keep track of, right, these complexes and other things that are all lumped together in my other models. Or you might do, right, a really detailed model. So this is one where I bind to DNA, I move along the DNA, I bind to the promoter, I go from an open complex to a closed complex to an open complex, I begin initiation, I then create mRNA one base pair at a time, right, et cetera, et cetera. In Biocompiler, you can more or less say, I want transcription at any one of these three levels, right? And so, and the same component, right, can generate models at different levels. So that's one of the things we've tried to build into Biocompiler. All right, so when we do all of that, this allows us to create models, and it allows us to create relatively complicated models when we want to in a relatively succinct way. So for example, you can't read what the different models are, but here's a bunch of different models. And what you see is the number of species in the model, number of reactions, the number of ODEs, and the lines of code. And what I want you to notice on this is that the lines of code is always small, right? So in fact, for all of these, we had 6 lines of code, 7 lines of code, 11 lines of code, 10 lines of code, 11 lines of code, and 14 lines of code, right? So that last model used 14 lines of code to generate a model that had 180 species, 337 reactions, and 587 ODEs that were used to describe all of that. Biocompiler does that automatically. You think at the level of the 14 lines of code, right? I want transcription, I want translation, I want repression, right? And then you say, now for the model I want to generate, depending on what problem I'm trying to solve, here's the mechanisms that I want to put into place. So Biocompiler is our tool that we're using to try and implement some of these ideas. And this is done by William Poole, who's actually here at this workshop, as well as Ayush Pandey, Andre Schur, Zoltan Tuza, and others. And so happy to talk more about it during the break. Okay, so our goal here is really to think about getting Biocompiler to be something that different groups can take those subsystems and model them and put them together. And that's actually something that we do within Build-A-Cell and that we do within my group, is to try and have different people model things and put them together and then go do the experiment and put them together and see what happens. Okay, so that's really what I wanted to talk about. Oh, sorry, and if you're interested, you can get all of this on the Build-A-Cell GitHub site. So that's what I wanted to kind of talk about today. So I've sort of told you about this picture of a synthetic cell, which is something that many people have kind of articulated. I think we're within 9 to 15 years of being able to create these. We have lots of individual groups working on the components, and I think that's good. And we need to figure out how to start connecting them together. I think there's some things that I don't see quite enough people working on, or maybe I see us struggling with and would love to get help. So chassis integrated sensing and transport, as I mentioned, locomotion subsystems, metabolism. Regulatory elements I haven't talked about. It's the area that we've probably worked on the most in my group. I think it's relatively well developed, but figuring out how to actually put the regulatory mechanisms in the subsystem so that they behave in the way that your models describe they should behave is open work to be done. Okay, so again, just this kind of notion that I think we need to go from talking about components to talking about circuits to really talking about systems and subsystems and integrating those subsystems. If this field is going to kind of be able to do things at scale, we're going to need to figure out how to get composability to work and how to get integration to work. I think we can move from individual cells to multicellular machines as we go through that process. Again, I would argue that synthetic cells, right, as opposed to putting things in living cells, is a more promising approach than cell-based methods. We'll see whether that happens or not. And my version of all of this working will be a synthetic machine programmed by engineers that can generate all sorts of different fruits out of things pulled out of the ground. Okay, and with that, I will stop and happy to go into one of the breakout sessions and answer questions and get more help from everybody.
Thank you, Richard. I think that's the perfect talk to finish off this session and the second day. I'm sure there will be a lot of interesting discussion resulting from that, and hopefully good suggestions to actually make this vision a reality.